Turns out, though, that there are not all of these G proteins are actually going to activate the adenylyl cyclase. So remember, we also had like our GIs, which are going to inactivate it. And therefore, they would decrease the cyclic AMP. We also have these subunits GQ that instead of acting on adenylyl cyclase, they actually act through a different pathway. They act using a different second messengers, IP3 and calcium. They're going to do this by activating a different protein, phospholipase C. So phospholipase C is basically a lipase is going to cut a lipid. What it's going to do is it's going to more specifically cut phosphatidylinositol 4,5-bisphosphate, or pt2, cut its kind of like head off, so that you're left with diacylglycerol or DAG and inositol 1,4,5-triphosphate, or IP3. So remember that in a phospholipid, we had this phospholipid head, or we had this phosphate-containing head group that was hydrophilic, and then this hydrophobic tails. If we cut that hydrophilic head off, well, now we have this soluble part, this IP3, this hydrophilic head is now kind of like free-floating. And we also have this part that's still embedded in the membrane, the lipidy part, the diacylglycerol. Now, both of these are going to serve important signaling functions. So we have a receptor, and this receptor, we still have a heterotrimeric G protein, and it still has an alpha subunit, a beta subunit, and a gamma subunit. But in this case, our alpha subunit is going to be one of these Q subunits. I tried to find what the Q stand for, but no luck. So instead of trying to go down a bunch of rabbit holes, I'm going to just leave it as it's Q. This GQ subunit, what it does is it's going to activate phospholipase C, or PLC, which is another one of those membrane-embedded proteins. So just like we had adenylyl cyclase that the alpha subunit was traveling to, this S-alpha subunit was traveling to, or the I1, with the phospholipase C, here we have the Q subunit traveling through the membrane, thanks to its kind of like lipid-attached tail, and therefore it's going to go and interact with phospholipase C. When it's in its active form, it's going to activate phospholipase C. Phospholipase C is going to cut the head off of the phospholipid. When it cuts that head off, remember you get those two parts, you get IP3 and you get DAG. Now, the DAG is stuck in the membrane, but it can move laterally, and so it can go and it can associate with a different protein kinase C. So basically, in its headless form, it likes to hang out with protein kinase C, or PKC. Now, PKC is another protein kinase, so you can imagine that it's going to have a bunch of important regulatory roles by phosphorylating and changing the activity of a bunch of different proteins, but it's not enough for it to be bound by DAG. It also needs a second signal in order to actually get activated. It needs another second messenger. So we could call this pt2, and we can call this IP3 and this DAG, these small molecules. We can call these second messengers, but we actually need a third second messenger in this pathway as well, which is going to be our calcium. So all of those are combined going to act as these little molecules that are going to help send the message throughout the cell. Protein kinase C, it kind of has this like dual secure two-factor authentication. You don't want it turning on prematurely or anything. So we say, okay, well, it's not just enough to bind to DAG. We also want to have it bind to calcium. And how are we going to get calcium? Well, remember we talked about how in our cells we have those like circa pumps, which are going to use energy to pump calcium into our endoplasmic reticulum against its gradient, so that we can maintain a high amount of calcium inside of this little compartment in our cells, that if we then open up a channel, we can release it into the cytoplasm. So we have one of those receptor-gated channels that is going to be activated by the binding of IP3. When IP3 binds, we open up that channel, we let the calcium flow out. When the calcium flows out, now it can go and it can bind to protein kinase C. And so protein kinase C is now, it has those two factors it needs. It has the DAG and it has the calcium. And now what's going to happen is it's going to be activated, and then it can go and it can regulate a bunch of other proteins. So we've had the G alpha, the GQ alpha was going to activate. So the receptor activates GQ alpha. GQ alpha swaps GDP for GTP. Now activated, the GQ alpha is going to travel and go and find phospholipase C and activate it. Phospholipase C is going to chop the head off of pt2 to produce DAG and IP3. DAG is going to stay in the membrane, and it's going to go and it's going to travel and it's going to find protein kinase C. Protein kinase C has the power to do a lot of regulatory stuff, but in order to do all that regulatory stuff, it needs its two-factor authentication. It needs both the DAG and the IP3, and the calcium. How's it going to get the calcium? Well, the calcium is stored in the endoplasmic reticulum. So in order to get released, the calcium receptor has to bind to IP3. IP3 is going to bind to this receptor and open the channel. That's going to lead to the outflux of calcium. Now we're doing facilitated passive diffusion out from the endoplasmic reticulum into the cytoplasm, where it can go and it can bind to protein kinase C and activate it. And then a lot of stuff happens. So basically, diacylglycerol, inositol triphosphate, and calcium have related roles as secondary messengers. And there's a lot of different pathways that are going to actually act through them. So not only do we have that, we have adrenergic receptors that are going to respond, but we can also have a bunch of other signaling pathways, angiogenin, oxytocin, vasopressin, serotonin, all sorts of different pathways that are going to act through this as a second messenger. But how is that calcium actually going to be able to activate things? So we saw how it can kind of like help activate our RTKs, but it also activates a lot of other things. And so just how we can have cyclic AMP, not just to activate adenylyl cyclase, well, these second messengers can activate multiple other things as well. And the way that they do this is often through this through this protein called calmodulin. Now, calmodulin has this shape that's going to change drastically in the presence of calcium. Calmodulin has this shape that's it's this EF hand, it's called. And so you can kind of think about it. You can think about like having your finger up and your thumb out. Here it's your first finger, but if you want to remember the F, you can think about your middle finger being up. But basically it's just to represent this structure where you kind of have those domains are going to be curled around the calcium. And when the calcium isn't present, you're going to get this long helix. And then when the calcium binds, it kind of like bends in half in a way. Bottom line, you change the shape drastically in the presence of calcium. When you change the shape of a protein, well, now you're going to change its binding interactions. And so this is going to modulate calmodulin's interactions with other proteins. There are a lot of proteins that are going to be regulated by calmodulin, including calmodulin-dependent protein kinases, or these CAM kinases, as well as all sorts of other proteins, including, look at this, cyclic AMP phosphodiesterase. So we can already see we have crosstalk between these different pathways, our cyclic AMP pathway, which was regulated by adenylyl cyclase, and our pathways that were regulated by phospholipase C and the release of calcium. So basically, we have these two main paths that we can go from a G protein coupled receptor. We could go to have our G protein stimulate adenylyl cyclase, produce cyclic AMP, activate pKa, activate other stuff. We could also have our G protein receptor coupled receptor have a G protein which activates phospholipase C, which is then going to lead to the cleavage of the pt2 into IP3 and DAG, which are then IP3 is going to go and it's going to cause the release of calcium. Calcium and diacylglycerol are going to together activate PKC. And not only do you have one calcium molecule, though, because it's one of those secondary messengers, we're getting that like amplification. We're releasing a lot of calcium. And so not all of it is needed to go and activate PKC. Some of it can go and it can bind to calmodulin. Binding to calmodulin causes it to change shape and interact with other proteins, including our CAM kinase. This can then go and regulate a bunch of other stuff. And that's just a start. There are lots of other GPRs that we don't have time to go into, but once you know the basic mechanism and logic, you can figure out what's going on in any of them. So remember that if you see one of these S's, this is going to be a stimulatory. It's going to stimulate adenylyl cyclase. And a classic example of this was our beta-adrenergic receptor, which is going to respond to epinephrine by increasing the production of cyclic AMP and activating pKa. We also saw how it could go. We can have a GQ subunit that's going to go and it's going to activate the phospholipase C and lead to the release of calcium as well as IP3 and pt2 and STAG. We also can have these inhibitory subunits, like which vasopressin binds to one of these receptors. It has this inhibitory subunit, which is going to inhibit adenylyl cyclase, lead to the less cyclic AMP being made and less pKa being activated. We also have receptors that are going to do things like bind to light and or that are going to respond to light or bind to odorants or sweet tastants and cause signaling in different ways. So we see that our senses are intimately tied up with GPCRs. There are about like 800 different GPCRs, and we don't even know what some of them do. But bottom line is they all kind of have the same logic. And they can respond to all sorts of different types of hormones and signals. When it comes to hormones, we can talk about different classes of hormones. So we can have some that are like proteins, like little proteins, like gonadotropin, thyrotropin, follicle stimulating hormone, that like FSH. We can also have peptides, so just like short little pieces of proteins, things like angiotensin and bradykinin, neurotensin, somatostatin, vasopressin. We can have amines, so like these small sort of amino acid like thingamabobs, or just basically groups with amine groups, little molecules with amine groups. So we can have like acetylcholine, we can have our epinephrine, our histamine, serotonin. We can have other things, opioids, rhodopsin, melatonin, all these various signals, which you can imagine that you would need a different receptor for each of these. But by then controlling what receptors different cells have, you can control what sorts of cells are able to respond to what signals. Like maybe you don't need all of your cells to respond to something like oxytocin or vasopressin or dopamine. Maybe your toes don't care that much about serotonin, but they care about other things. And so the way that they care or don't care is because of what receptors they have and how many receptors they have and how things are scaffolded to that receptor. And that was just our GPCRs. So now let's talk about our second main type of kind of these receptor protein signal transduction machines thingamabobs, are RTKs, or receptor tyrosine kinases. In the case of our G protein coupled receptor, remember what our receptor was? It was that 7 transmembrane protein that was going to not be an enzyme itself, but it was attached to an enzyme. It was attached to that G protein that could ultimately act as a GTPase and then activate itself, act as that switch. And the role of the receptor was actually as one of those guanosine exchange factors to get that G protein to dump its GDP and form and pick up a GTP, activate that and cause the signaling. In the case of our receptor tyrosine kinase, here our receptor is itself an enzyme. It's going to be a tyrosine kinase. When we talk about kinases, basically the name before the kinase, the amino acid before the kinase is telling you what it phosphorylates, and so it phosphorylates tyrosines. More specifically, these receptor tyrosine kinases, they can actually phosphorylate themselves. And they can phosphorylate other things as well. But in order to be activated, they have to first phosphorylate themselves. And when something's phosphorylating itself, we call it autophosphorylation. So it actually phosphorylates like the other copy of itself. So these are going to receptor tyrosine kinases are going to act in like dimers. And so sometimes we'll see that they're going to be constitutive dimers, such as in the case of our insulin receptor, our INSR. It's going to act as a constitutive dimer. So it's always a dimer, but it's not active. On the other hand, we have other sorts of receptor tyrosine kinases that are going to be monomeric, and then only upon the binding of a ligand do they come together. So with our insulin receptor, basically here, the binding of a ligand activates it, but it doesn't cause the dimerization. It's already a dimer. But with these other ones, binding of a ligand causes the dimerization and then the activation. Once you have the activation of one of these, basically they autophosphorylate one another, then they start phosphorylating other proteins, leading to a kinase signaling cascade. How do you actually activate these? Basically what happens is, in the case of the insulin receptor, you have this dimer. So remember, it's this constitutive dimer, but it's going to have an inactive kinase domains. These tyrosine kinase domains are going to be capable of phosphorylating tyrosines, including tyrosines on themselves or on the other kind of like monomer, the other copy of itself. But they can't do this unless they kind of have the right conformation. And they won't have the right conformation unless you actually have the binding of the ligand. So the binding of the ligand causes a conformational change that's going to activate the subunits to activate one another. So basically it changes the shape of these subunits so that they're then able to go and phosphorylate one another. Once they phosphorylate one another, they basically phosphorylate this key part called the activation loop. This activation loop was blocking the substrate binding site. So the part where kind of like the active site where the substrate needs to bind to get phosphorylated, you have this activation loop that's blocking it. If you phosphorylate the activation loop, well, now you move that loop out of the way and allow substrate to bind. So these dimers, these monomers can basically phosphorylate one another's activation loops to move those loops out of the way and now cause the substrate binding site to be open so that they can phosphorylate things. We'll see that it can then go and it can phosphorylate this protein called IRS1. Now, IRS1 is basically then going to do, what it's going to do is it's going to, once it's phosphorylated, it's going to serve as sort of this kind of connection point. So IRS1 stands for insulin receptor substrate 1. And what it does is it's kind of just this hub. So it has these sites that get phosphorylated by the insulin receptor, but only remember if the insulin receptor is activated. So if the insulin receptor binds to insulin, it activates itself, which is then going to go and phosphorylate IRS1. Once IRS1 is phosphorylated, well, now it's going to have a binding site for a protein called GRB2. And GRB2 is basically then going to bind to this protein called SOS. SOS is then going to bind to this protein called RAS. And what's RAS? Well, RAS is one of our G proteins. And so RAS is one of those little G proteins that's basically, this one's monomeric, but it's able to do just what all G proteins do. It's able to take GTP and break it down. And it's able to then exchange that GDP, the broken down the end product of that breakdown, the GDP, it can dump that and bind to GTP. But remember, that's going to be a slow process. And so we can have proteins that are going to speed things up. And one of the proteins that can speed things up is going to be this SOS protein. This SOS protein is then therefore going to act as one of those guanosine exchange factors. It's going to help RAS switch out the GDP for the GTP and activate RAS. The activated RAS, well, it is going to bind and activate RAF1. Now, RAF is going to be a kinase, and it's going to set off a signaling cascade of kinases. So when we have a kinase, and we have one kinase and it can phosphorylate a lot of stuff. What if we have a kinase phosphorylated kinase? Well, now we can have that one kinase phosphorylate a lot of other kinases, which can phosphorylate a lot of other kinases, which can phosphorylate another kinases, which can phosphorylate a lot of other kinases. And so in this way, we're going to get a big signal amplification, even without the production of any really like secondary messenger. But we can have this phosphorylation cascade where ultimately we can phosphorylate a protein that can then go into the nucleus and affect the transcription of genes. And so this is one of the common pathways that we'll see is this like MEK pathway. So we had ultimately this protein ERK that was the one that was actually getting into the nucleus. And it turns out that ERK, as well as MEK, so the thing that activated ERK, as well as RAF, which was the thing that activated MEK, these are all basically in larger families. And these families have kind of ridiculous names. So ERK is in the MAPK family for myogen activated protein kinases. And it's specific for serine or threonine residues. So it's not a tyrosine kinase. instead of phosphorylating tyrosines, it phosphorylates serine to 3 residues. And not just any ones, it has like all kinases, they have like consensus sequences, which are the things that they like to phosphorylate. So they're not just phosphorylating anything. But it phosphorylates specific serine or threonine residues. And it can phosphorylate things like transcription factors, such as ALK1, which is then going to activate them and cause differences in the transcription of different genes. And often for like insulin, you'd have things that were needed for cell division. But ERK was activated by, so if we had ERK be in this MAPK family, well, it was activated by MEK. And MEK is in the MAP kinase family. So basically, it's a kinase that activates the MAP kinase. And so MEK is going to be in the MAPKK family. And then if we had a MAP kinase that was activated by another kinase, well, that other kinase would be a MAP kinase. So RAF1 is in the MAP kinase or MAPKKK family. And so basically, yeah, the naming can get complicated. But basically, what I find is the most useful is just kind of being able to look at pathways and look at arrows, what's going where, and not try to memorize everything. Speaking of what's going where, how do things know like where to go? Basically, if you imagine how many different proteins are in a cell, you're going to need a way to kind of hold things together. And one of the ways that you'll do this is with these scaffolding proteins. So this is one example, this kinase suppressor of RAS. Basically, this is a scaffolding protein that's kind of going to hold together all those components of the pathway so that things can find each other more easily. How do these things all kind of like bind together? A lot of it is going to be mediated by these, by binding to phosphorylation sites. So there are a couple of main kind of like phosphate binding domains. So one is this SH2 domain, which is a protein domain that binds to phosphorylated tyrosine residues. We also have SH3 and pH domains, which are protein domains that bind the membrane phospholipid PIP3. So we have phosphorylation mediated ways in which we could kind of bind to membranes and which we could bind to phosphorylated proteins. And what you see a lot is that basically different types of proteins are going to have different combinations of these SH2 and SH3 domains, as well as a lot of other domains. And so by kind of like having all of these different domains and all of these sites, you're then able to build these kind of places where these proteins can interact with one another in specific ways that are going to be mediated by phosphorylation. So though we talk about like the SH2 domain as being like binding phosphorylated sites, remember that it's not only just that one phosphosite that's going to be important for distinguishing between different proteins, there's going to be a lot of interactions. But by having this one like place where it can kind of recognize phosphorylated versus not phosphorylated, you can therefore regulate when things are bound where. So you bring things together, say only when they're in the phosphorylated or the not phosphorylated form. So RAS, remember we said RAS is one of those G proteins. So in this signaling pathway of insulin signaling through RAS, we are using a G protein, but we're not using a G protein coupled receptor. Our G protein is not directly attached to our receptor, and it's monomeric instead of being heterotrimeric. So you can see that basically RAS is down the line from our receptor, connected through the IRS1, which is bound to GREB2, and which was bound to SOS, which was going to be bound to our RAS. Now, RAS you might have heard about because it's going to be very important in the context of cancer. Mutations in RAS that like eliminate its GTPase activity are going to cause it to remain constitutively active or constantly active. These sorts of mutations occur in about 25% of human cancers. You can also get mutations in gaps. So those proteins that were actually going to cause the G protein to activate its GTPase activity. So basically the mutation in RAS itself can alter the natural GTPase, like the intrinsic GTPase activity of the G protein. But you can also have mutations in the regulatory proteins that regulate the G proteins, such as this tumor suppressor gene, NF1. So NF1 encodes a gap, which causes RAS to stay active for an extended period if you have a mutation that's going to inactivate that NF1. When we talk about a tumor suppressor gene, basically that's a gene whose normal function is to kind of like keep a check on cell growth. If you have a mutation in one of those, well, now you're going to have, you're removing the brakes. There are also proteins that we can call like oncogenes or proto-oncogenes. If the normal gene would be a proto-oncogene, and then when it's mutated, it becomes an oncogene. And these sorts of genes are things that are going to kind of stimulate growth, division, this sorts of thing. So that was one pathway that we could go with the insulin receptor, where we had this adaptor proteins, the RAS activating protein, the RAS, the MAP kinase, the MAP kinase, the MAP kinase, and then into this nucleus and affecting things. But we also have insulin receptors or other types of protein tyrosine kinases, tyrosine kinase receptors that are able to actually act through a different pathway. And so they can actually act through that same phospholipase C pathway that we saw before. Additionally, insulin can act through this pi3K pathway. Basically, in this case, we still have our key player, this IRS1. Remember that IRS1 was getting phosphorylated by our insulin receptor. Here, once phosphorylated, it associated with this GRB2 kind of like scaffolding protein, which is associated with SOS, which activated RAS. In the case of this other pathway, here what's going to happen is that phosphorylated IRS1 is going to interact with this PI3K. So IRS1, once it's phosphorylated, well, now it can bind to PI3K. When you see a K, think kinase. This is going to be a kinase. It has an SH2 domain that's able to bind to the phosphorylated IRS1 and get activated. Once activated, well, here it can basically phosphorylate pt2. pt2 is phosphatidylinositol 4,5-bisphosphate, which is one of our phospholipids. PI3K is basically able to phosphorylate that to give you PIP3. Don't confuse this with IP3. This is still like a full phospholipid, but it has a third phosphate group on this phosphatidylinositol head. And once it's phosphorylated, well, now it can bind to PKB. Another K, another kinase, we've got protein kinase B. Protein kinase B is going to bind to PIP3, and it's going to be phosphorylated by another kinase, which is called PDK1. And then this is going to be fully activated. Now, this PKB is going to phosphorylate a protein called GSK3. Why do we care about GSK3? Well, GSK3's normal role is to regulate this glycogen synthase. And so glycogen synthase making glycogen, remember that's our storage unit for glucose. So it can kind of build glycogen from glucose and help you store glucose. Let's think about what would happen if we had insulin. When do we have insulin released? Well, we have insulin released if we have had a big meal or we have a bunch of glucose. Basically, it tells our cells to let in that glucose and starts like making and like storing it and using it and things like this, or I guess storing it. We wouldn't want to basically break down glycogen to make more glucose if there's plenty of glucose around. Instead, if there's a lot of glucose around, we're going to want to store that. And so we're going to want to make glycogen in the response to insulin. So in the response to epinephrine, remember we said we wanted to kind of like break down glycogen. In the response to insulin, we're going to want to build up glycogen. So what we're going to want to do is we're going to want to have our glycogen synthase be active. Glycogen synthase is going to be regulated. Its activity is regulated by this kinase called GSK3. GSK3, when it's active, it's going to phosphorylate glycogen synthase. So I know that a lot of times we talk about phosphorylation as activating things, but in this case, phosphorylation is actually going to inactivate our glycogen synthase. Once it's inactive, it can't make glycogen. So if we have GSK3 active, well, our glycogen synthase is going to be inactive. But if we inactivate GSK3, well, now we prevent it from inactivating GS, which means we can make glycogen. So PKB, what it's going to do is it's going to inactivate GSK3, and therefore it's going to turn the brakes off of the brakes on the glycogen maker. So it's like a complicated pathway, but at the end result, you get glycogen made. Also, what happens is that PKB, remember it's a kinase, it can phosphorylate it doesn't just have to phosphorylate a single thing. It's an enzyme. It can phosphorylate over and over and over again, and it can phosphorylate different molecules. And so each kinase is going to have like specific molecules that'll phosphorylate. They have to have like the consensus sequence and everything, but they can still be multiple proteins that they're going to phosphorylate. In the case of PKB, one of the proteins that it's going to be able to act through are going to be other G proteins. And so we have these, you can't escape those G proteins in signaling. In this case, we have RAC1, RC1, and RAB. Basically, long story short, what they're going to do is they're going to cause the movement of these GLUT4, these glucose channels, from these like internal membranes to the surface of the cell, allowing glucose to be taken in. So basically, if you want to if you think about it, if you want to be able to take glucose into like regulate how glucose gets into your cells, one way is by regulating how much of the glucose receptors and transporters are on the surface of the cell. But actually going and making those proteins and those transporters is going to take time. If you want to be able to respond more quickly, you're going to want to have them pre-made. You can kind of like have them pre-made, but kind of like held inside the cell in these little kind of membrane pouches. And these internal vesicles can then merge with the membrane once they get the signal, and the signal is going to be relayed through these G proteins, allowing you to move those transporters to the surface and let glucose in. So those was just, we saw how insulin could lead to these different pathways, and we can also have lots of different receptor tyrosine kinases. So again, insulin's kind of weird, or insulin receptor is kind of weird in that it's kind of a constitutive dimer, but most of these are going to be monomers usually. Some examples are these growth factor receptors. VEGFR and this sort of thing, EGFR, various receptors. And these are, a lot of these are important for controlling cell division. So it's really important that cells tightly control their cell division, or if not, basically you get uncontrolled cell growth, which is cancer. So there's different phases of cell division to cell division, and basically there are different checkpoints at these different stages so that cells like only divide if when the time's right. But if there are problems with that, then you can get missignaling, you can get uncontrolled growth. One way in which cells kind of control things is through phosphorylation via cyclin-dependent protein kinases, or CDKs, which are made in response to growth factor-based signaling. So you have these growth factors that are going to lead through receptor tyrosine kinases to one of those like MAPK cascades. They cause things like the phosphorylation of these proteins Jun and Phos in the nucleus, which are then going to activate this transcription factor, which are going to regulate the making of those cyclins and those CDKs, enzymes for DNA synthesis, and cause the cells to go from from G1 to this S phase. So basically here in this S phase, they're making that DNA, they're preparing to divide. And so basically these signaling factors are going to help tell it to divide. And you can imagine that you could have mutations in all sorts of different places that could therefore cause this to happen. And so basically there are all sorts of different types of receptor tyrosine kinases, and they'll have different substrate specificities. So they'll bind to different types of primary messengers. Some of them actually don't have like a known ligand, or some of them we don't think they actually have a ligand at all. And so there's this one called HER2. And so basically HER2, you might have heard about it in the context of like breast cancer. HER2 is one of those ones that doesn't need a ligand to activate it, but it does need to form a dimer. And so normally you don't have a lot of HER2. And so you're not forming any of these like many homodimers. But what happens, so those homodimers, they wouldn't need a ligand to be activated. But what could happen is basically often this HER2 is going to team up with another RTK, which did need a ligand. So you still had this ligand activation. In the case of overexpression of HER2, however, so if cells are making a lot of it, such as cancer cells that overexpress HER2, well, now they're able to make a lot of this functional dimer that doesn't need a ligand to be activated. If it doesn't need the ligand to be activated, well, now it's going to be like constitutively active if there's enough of it where you form this dimers. So that's one of the common ways in which one of the common mutations associated with breast cancer is like an overexpression of HER2. And there are antibody treatments like Herceptin that actually go and try to block this receptor so that it can't be activated. You can't get that activity. There's also a lot of crosstalk between the pathways. So that insulin receptor, it can also go and like that, it can phosphorylate the beta adrenergic receptor. IRS1, that was acting through PKB, which could phosphorylate the beta adrenergic receptor. All those things that are basically able to kind of control the internalization of those receptors. But you can also have it's basically the insulin receptor can phosphorylate a different site on the GPCR, which can then lead it to kind of be a building off point for one of those MAP cascades. So lots of crosstalk between the pathways, which a lot of it depending on kind of like what's hooked up where. But that is basically a broad overview of signal transduction. We have those GPCRs where we have the G protein coupled receptors bound to a heterotrimeric G protein. The G alpha subunit is the one that's going to bind to GDP and exchange it for GTP upon response to the primary messenger that's the signal relayed through the receptor. So the primary messenger, the hormone, is going to bind to the receptor, cause the receptor to change shape. The change shaped receptor is then going to basically cause the G protein to change shape to release its GDP and take up GTP. Once bound to GTP, well, now it kind of exposes a binding site for adenylyl cyclase. And so it's going to ditch that those beta and the gamma subunits, go and find the adenylyl cyclase and activate it. Adenylyl cyclase is then able to produce cyclic AMP, our second messenger, which can then go activate things like protein kinase A, which can activate other things. Alternatively, it can go in and can activate protein lipase C, which is basically going to lead to the production of IP3 and DAG, which are then going to act through a different pathway. We can also have our receptor tyrosine kinases. Here, these are going to function as dimers. The ligand binds, activates autophosphorylation. Those dimers are going to basically phosphorylate one another and lead to signaling cascades through kinases typically, as well as pathways that can act through other lipid-mediated functions. We also have our gated ion channels where things are going to open up in response to a stimulus. And we have things like steroids, which can just go directly into the nucleus and bind to receptors there to alter transcription. Long story short, this was a lot of detail, a lot more than you need to know. But if you just wanted some cool facts about biochemistry, that's that. There's still a lot more that I didn't go into. But basically, by kind of having an idea about the general logic, you can then go and look at pathway diagrams and kind of try to figure out what's going on and what might happen if you have mutations at different points, if you have agonists or antagonists, places you might want to see regulation, and then all that good stuff.